Good afternoon. I think Netta's chime is going to put us all in a post-lunch lull. So we're going to get started. I'm Kara Meister. I'm an assistant professor in pediatric otolaryngology and on the MCHRI education committee. We're good? You're okay? Okay, great. This session is truly my honor to moderate. So as Dr. Leonard shared earlier, one of the goals of the MCHRI is to educate and promote and prepare the next generation of researchers and investigators. And now it's our turn to learn from 5 of the truly best and brightest young scientists, and they're going to share with us their groundbreaking research. If anyone is in the audience and feels inspired by this morning or this afternoon or any of the amazing posters that you've been able to see, please check out the website. There's a lot of funding available to you as well as other resources. So our first speaker is Amanda Dudek. Amanda received her BS from Colorado State University in 2018 and defended her PhD in virology from Harvard, where her dissertation focused on cellular entry mechanisms of adeno-associated viral vectors. For this, she received the Excellence in Research Award from the American Society of Gene and Cell Therapy. Since 2019, she has been a postdoctorate fellow in Matthew Porteous' lab before being promoted to an instructor this year. Congratulations. Her research focuses on the use of gene editing for HIV therapy development and understanding HIV replication in human T cells. Amanda.
Thank you. Hello? Thank you, Kara, for the introduction and to the organizers for selecting me to speak today. I'm excited to tell you about a project I've been working on in the Porteous lab. So we primarily use gene editing to develop therapies for blood and immune diseases, including HIV. But today I'm going to talk about a side project that I've been working on using some of these methods to really turn back on basic biology and try to understand control of virus replication. So for most patients that are infected with HIV, their immune system is not able to adequately recognize and destroy the infected cells. So in the absence of therapy, they have high viral load, low CD4 cell count, and progression to AIDS. They're able to transmit the virus horizontally between adults and also vertically from mother to child, either in utero or during childbirth. However, when on antiretroviral therapy, many patients are able to reach undetectable levels of virus in the serum, and it's generally thought that undetectable means untransmissible. However, there's also a rare patient population termed elite controllers or long-term non-progressors who are able to effectively recognize and control all the infected cells, leading to not requiring antiretroviral therapy, but achieving lower undetectable viral load throughout their lifetime. However, these patients can still transmit virus both horizontally and vertically. And as a drastic oversimplification of something that's still really not fully understood, this is essentially thought to be due to the efficiency of MHC presentation of viral peptides on the surface of infected cells, as well as the strength of the innate and cytotoxic response to the infected cells. So HIV actually does this very strange thing where, if you can see in the red box, HIV positive cells downregulate canonical MHC molecules and actually upregulate on the bottom a non-canonical MHC called HLA-E. This group also showed that in K562 cells overexpressing HLA-E, in the white bars you're able to see efficient cell killing by natural killer cells. However, there is a very specific HIV-derived peptide from one of the structural capsid proteins, which is able, in the red arrow, to inhibit NK cell-mediated destruction as efficiently or more efficiently even as self-peptide. So this suggests that HLA-E may actually be advantageous to HIV escape of immune recognition. However, there's really a disconnect between these clinical observations and the mechanistic explanations that we are able to find in quite honestly irrelevant cell lines. So I'm wanting to see if we can prove this mechanism in true primary human immune cells and repair this broken link in our chain of understanding. So to do this, I'm using a Cas9 RMP delivery into peripheral blood-derived CD4 positive T cells, and if we are able to adequately express the receptors and co-receptors on these cells and infect them, we can repair this link. So when I first started the project, showed that essentially we see almost no expression of the required co-receptors on these T cells. However, after a lot of optimization, I was able to find conditions where we can upregulate the co-receptors. And on the top, I'm showing just control-treated cells, and on the bottom, these are cells that are edited to knock out CCR5, showing that we see a loss of the surface protein. This is a much more complicated diagram than you need to know, but just to show you all of the details if you're curious. On the left side, essentially the model I'm using is to isolate T cells, activate, edit, differentiate them so that we can infect them. And then on the right side of this diagram, we take donor-matched natural killer cells and either allow the infection to continue with T cells alone or add them in with the T cells in a co-culture method. So on the top, I'm showing very efficient knockout both at the genome and protein level of HLA-E, but importantly, the HIV receptor, CD4, is not changed, as well as neither of the HIV co-receptors are lost in the HLA-E knockout. So after we look at a replication-competent infection in our T cell or co-culture system, in the black lines, I'm showing the drop in virus concentration in the supernatant with T cells alone versus NK cells. And we do see a slight drop in infection. However, importantly, in the two red lines, we see that when we knock out HLA-E, we see a much more drastic drop in the concentration of virus in the supernatant, suggesting these cells are able to control virus better when HLA-E is not there. And this is just showing the same experiment in another donor to highlight a bit of the patient-to-patient variability. However, I've tested now in 5 independent, healthy human blood donors, and all of these donors show that there's an increase in control in the presence of HLA-E knockout. However, we see drastic differences in the efficacy of this, some donors showing only about a 2.5-fold drop in virus concentration, and some donors showing up to an 82-fold drop in concentration. And so my next step is really just to try to understand the natural killer cell side of things and see if we can understand this mechanism of control a little bit better. And I just want to thank you all for listening. Thanks to my lab and my friend and collaborator, Alex Cocker, who was the mastermind behind this project, really. And I'm happy to take any questions.
Great, an immense body of work, and truly an element of team science. Any questions from the audience? We have time, I think, for 1, possibly 2. I don't know much about HIV, but I will ask, and I think you already alluded to this, what might explain the difference of why some donors had a 2.5 and some all the way to an 8-fold increase?
Absolutely. So it was actually up to 80-fold decrease in some patients. And what I'm starting to look at is on the last slide, I kind of showed there are these activating and inhibitory receptors on natural killer cells, and these two highlighted here are actually specific for HLA-E interaction. And what I've started to look at is the ratio of these receptors, and we think that may be playing a role, but also, at least in the context of elite controllers, there are certain SNPs in those proteins and or certain HLA-E subtypes that we think are actually just increasing the activity of this kind of interaction and the killing.
Very interesting. One more?
Thank you. Cool work.
Thank you.
I'm wondering if you isolate the CD56 NK cells, I guess, right? And do like an autologous culture. So there are CD56 bright and dim cells, do you look at those? Because if you have an overrepresentation of the cytotoxic ones, that might maybe influence your readout there.
Absolutely. So, so this was just a bulk NK cells. I do want to look at the subsets. Actually, 56 negative NK cells are expanded in chronic infection, like with HIV and also CMV. So I do want to look at these different subsets. And actually, the 56 negative subset, we do. We need to look at more donors, but there does seem to be a correlation of the A to C ratio on the 56 negative cells that correlates quite well with the fold decrease in infection.
Great. We're going to hold the next set of questions and move on to the next speaker, but please keep it because we hopefully built in a buffer at the end. Our second speaker is Xiwei Shi, a postdoctoral scholar in the Department of Neurology. He received his BS degree in computer science from Shanghai Zhaotong University in 2013 and an MS degree in biomedical engineering from Xiwei University in 2016. He worked as a research assistant at USC Neural Modeling and Interface Laboratory and received his PhD degree in biomedical engineering from the University of Southern California in 2002. After graduation, we were lucky to recruit him at Stanford University as a postdoctoral scholar in the Pediatric Neurostimulation Laboratory Bomber Lab, and he's a member of the Xiwei Institute. His research interests are largely directed towards identifying the causal relationship, hopefully not casual, causal relationship of neurons and brain regions, and understanding how information is encoded in neural signals by employing machine learning modules. Welcome.
Thanks for the introduction. Good afternoon, I'm Xiwei, a postdoc from the Bomber Lab. I'm also a recipient of the MCHRI Postdoc Support Award. So the title of my presentation is Repetitive Transcranial Magnetic Stimulation Modulate Brain Connectivity in Children with SLACs. Children with self-limited epilepsy with centrotemporal spikes is a common pediatric epilepsy, accounting for 15 to 25% of all pediatric epilepsy syndrome. And children with SLACs have moderate difficulties with language and have been found with atypical development of their language networks. Patients are usually found with experience interictal abnormal epileptic spikes, which has been known will disturb cognitive function. And our previous study suggests that those spikes are associated with increase and diffuse of increase of brain connectivity in children with SLACs comparing to controls. And repetitive transcranial magnetic stimulation is a non-invasive tool for modulating cortical excitability, and it has been found that low-frequency RTMS can yield inhibition effects to their excitability. Therefore, in this study, we are interested in investigating whether low-frequency RTMS can modulate brain connectivity in children with SLACs. And to do this, we deliver 1 Hz RTMS to the motor cortex and measure the connectivity between brain regions before and after the RTMS. Our regions of interest are motor, frontal, and temporal, which are regions that are associated with language development. And our experiment setup consists of 2 days of RTMS. On each day, the participants will receive 1,000 pulses of either real or sham RTMS to the motor cortex. Before and after the RTMS, we deliver single pulse TMS together while the EEG signals were recorded, so that we can measure the connectivity between brain regions before and after RTMS. So far, we have recruited 17 children with epilepsy participate in this study, and here are some of our results. So the left subplot shows the distribution of connectivity change after the RTMS. The blue boxes are the change after real, and the orange ones are after sham. The stars marks out the region that had statistically significant change to the baseline after the RTMS. And the right plot is just another different visualization showing the significant change regions after RTMS in a brain mapping. So as we can see that all regions had connectivity reduction after real RTMS, but not after sham. Also, we want to point out that although some regions are not statistically significant, we actually see a widespread connectivity reduction in all of our regions of interest. And then we next test how's the effects of RTMS on the frequency of spikes, which have been known to be will abnormally increase the brain connectivity. So we had 5 subjects who had even lots of spike during the experiments while the EEG were recording, and we counted the number of spikes from those 5 subjects before and after a certain condition of RTMS. And our results show that after real RTMS, the frequency of spike gets lower. However, the frequency of spike doesn't change much after sham RTMS. And this is another visualization showing the absolute change of spike count before and after real or sham RTMS. So, so far we have shown that real RTMS can reduce connectivity overall, and also RTMS can reduce the frequency of spike. So we next want to investigate whether the overall reduction of connectivity was mainly driven by the reduced number of spike, or RTMS had any effects additional to that. To do this, we did similar connectivity comparison. However, in this case, we remove all the spikes and only check the connectivity before and after in spike-free period. And our results show that although less significant, we still see a widespread reduction in all connectivity, in all regions of interest after real, but not after sham RTMS. So this result together suggests that real but not sham low frequency RTMS can modulate brain connectivity in children with epilepsy by reducing the frequency of spikes and by reducing the abnormally increased brain connectivity in spike-free period only. Also, so we believe that low-frequency RTMS provides as a potentially therapeutic tool for modulating abnormally increased brain connectivity in children with epilepsy. And thanks for listening, and thanks to all our collaborators for their great helps, and also a special thanks to MCHRI for supporting our research. Yep.
Great. It's easy to see how that might translate to actual patient benefit. So,
Yeah, great.
Dr. Oro.
Hi, Tony Oro. Wonderful talk. I love the technology. So it's something shows that RTMS has variable remission. Yeah. Do you have a sense, like if you check them later to see how long the effect happened or predicted it, and how often you'd have to do this therapy to keep the spike production down?
Yeah, that's a great question. So far, we only have 2 days that's apart with 1 week interval. And we mainly compare the effects of real and sham RTMS. However, we also propose to do, recruit new patients to do a 5 days crossover sham-controlled experiments to see how's the accumulative effects or RTMS, and as you said, how long that the RTMS can last for their effects. Yep.
And do you think it's the same or different than depression or chronic depression or pain?
Well, I cannot guarantee anything, but we do see some variance about the RTMS effects. However, on group level, as we show here, like across 17 subjects, we see that the direction is inhibition, like reduce the connectivity. And there are some literature also support such inhibition effects, but I do agree that the effects can be variant and depends on each individual.
Xiwei, I have to ask, how do you perform sham RTMS?
Sham RTMS is performed by you flip the coil and you patients hear the sound and feel the clicking. However, the actual TMS was not penetrating your skull. Yeah.
Okay, excellent. Thank you very much. Our middle speaker, third, is Leah Schwartzrock. Leah is a research professional in the lab of Dr. Anishka Shekowitz, studying hematopoietic stem cell biology, transplantation, gene therapy, gene editing, and regenerative medicine. She received her bachelor's degree in 2018 from Iowa State University and joined us in 2020 by way of Mass General Hospital. Her current projects explore treatments for bone marrow failure, syndromes including Fanconi anemia, which she will speak about today. Leah is motivated by a passion to help others and contribute to innovative science that will make advancements towards safe and effective therapies in these patients. Leah.
Thank you. Today I'm going to be talking about in utero hematopoietic stem cell transplantation for Fanconi anemia. Fanconi anemia is an inherited autosomal recessive genetic disease that is caused by defects in one of the 23 different FANC genes. The most common are in FANC-A, FANC-C, and FANC-G. The FANC proteins contribute to the FANC complex, which is important for DNA damage repair. Patients are diagnosed by abnormal chromosome breakage studies, which can be done on blood or fibroblasts, and subsequent FANC sequencing. And then this can be done postnatally or in utero via CBS or amniocentesis. Fanconi anemia causes progressive bone marrow failure in almost all FA patients, with 80% having bone marrow failure by the age of 12. It is also a systemic disease with all tissues having sensitivity to DNA damage. These patients have a greater than 1,000 times increased risk of malignancy than normal individuals. Allogeneic hematopoietic cell transplantation, or HCT, is curative for the hematologic disease, and it has good and improving survival, but it is a resource-intensive treatment with both short-term and long-term toxicities. And just to take a moment to highlight the graphs here, you can see that the overall survival has improved, but FANC-A patients that have been transplanted have a much higher risk of solid tumor after transplant. So our lab wanted to ask the question, can we do better and create a preventative treatment that can be given in utero? So some of our rationale behind in utero HCT is that given the fetal immune system, there should be no need for immune suppression. Given that the HSCs in FA are failing, wild-type HSCs may be able to outcompete the diseased HSCs, and therefore you won't need myeloablative conditioning. It could potentially enable a safe and preventative treatment of the hematologic and immune disease for FA. And additionally, it has potential for rapid translation given similar concurrent in utero HCT trials. This is already being done for alpha thalassemia major as part of an FDA-approved phase 1 clinical trial open at UCSF. And that's being led by Tippy McKenzie, and they're giving maternal bone marrow via the ultrasound-guided umbilical vein injection. And 6 patients have been treated to date, which shows the safety, but they haven't seen much efficacy or engraftment, and that may be due just to the revved-up environment of the alpha thal bone marrow versus we believe the FA bone marrow, since it's failing, may be a more ideal indication. So to test this, we set up preclinical experiments in mice. We took FANC heterozygous and mated them together so we can date the pregnancies, and then we took whole bone marrow from CD45.1 donors and enriched for c-KIT or CD117. And we then injected fetuses at E13.5 to E14.5, allowed the pups to be born naturally, and then genotyped and analyzed the donor chimerism. So our initial results in our FANC-D2 model were very exciting. We saw that a very robust engraftment in our homozygous mice that continued to increase with time, reaching almost 95% by 30 weeks. And then compared to the heterozygous and wild-type littermates, which plateaued around 3 to 4% and consistently had the same engraftment there. And we could see this across multiple lineages in the peripheral blood. In our FANC-A model, we're starting to see a similar trend, more robust engraftment in the homozygous that is increasing with time across the lineages, although this may be a little less robust due to the FANC-A phenotype being a little less severe than the FANC-D2. We checked the bone marrow and saw promising long-term engraftment in the HSCs, and leading us to believe the high donor engraftment is likely due to the fetal immune tolerance and competitive advantage of the wild-type cells. So in conclusion, we've seen high donor engraftment in both of our FANC models with in utero HCT. We have not seen any immunologic rejection. We've seen increasing engraftment with time, showcasing that competitive advantage, and are continuing to do functional studies to see if there's correction of the DNA damage deficiency. We're all additionally working with colleagues, specifically the CDCM and the Dunleavy Maternal Fetal Medicine Center, to help translate these results to FA patients, including the initiation of a clinical trial. And we also hope this would justify patient care for FA patient care advocacy and understanding of the disease. I'd like to thank our sponsors and my lab for their help on this project, and I'd be happy to take any questions.
Really exciting, truly groundbreaking work. As a head and neck surgeon, I take care of a lot of patients with Fanconi anemia to screen for solid tumor, and the burden of this disease is really incredible for the whole lifespan. So kudos to trying to solve it. Question?
It's very exciting work. So you are following up to see if they get secondary cancers. And what about the other tissues that are not hematopoietic? They still going to have defects. Have you looked at that?
Yeah, so this is very preliminary data. These mice are actually still alive. But our current plan is to look at all tissues and specifically look at immune cells in those tissues to see if we've gotten donor engraftment that may help with the surveillance and ability of getting tumors in those areas.
And the transplanted hematopoietic stem cells, they're showing normal radiosensitivity and chemotherapeutic sensitivities. Have you tested those cells?
We haven't tested them yet, but the plan is to give them DNA damaging agents like MMC, where we can do, and test that, yes.
Hi, Tony. Great talk. Can you just talk about in terms of like the entrance requirements for a potential clinical trial? Would you, because of the bone marrow chimerism you get, so would you take then the B2, not the A? And do you think that's why TP didn't see any good effect for alpha thal patients because the chimerism didn't work? Or can you maybe talk about that?
Yeah, I can't speak too much about the alpha thal, but I know they are using currently maternal CD34 positive cells. I think that the field, and maybe Anishka can speak more to this as a clinician, is moving more to alpha beta depleted grafts. And I also think the alpha thal environment is just more, it's trying to create, more blood cells. It's like doing the opposite of FA in the sense that where it's not being depleted by the bone marrow failure.
Okay. Excellent. The fourth groundbreaking young investigator in this session is Feng Xie, currently a postdoctoral scholar at Stanford University School of Medicine. He holds a joint PhD from Duke University and the National University of Singapore. He previously obtained his bachelor's degree from Tsinghua University in Beijing in 2017. During his PhD time, he utilized interpretable machine learning tools in acute and emergency care settings and published 8 first author research papers in very impactful journals with a total impact factor of over 60. Specifically, he has created novel informatics framework called AutoScore, which generates interpretable clinical scores from the EHR. It's an open source software that's been used by local and international researchers, and it's been downloaded more than 10,000 times. The first paper was published in 2020, and to date, it's generated over 60 official citations. His research interests include machine learning, clinical informatics and decision-making, predictive models, the EHR, and stratification of risk in acute care settings.
Okay. Thanks very much for the very kind introduction. And good afternoon, everyone. I'm Feng Xie from Nima Agai Post Lab. And I'm very happy to share our research in predicting neonatal disease using machine learning and large language models. And first, let me introduce some previous research from our lab. Our lab has a broad interest in maternal and children's health plus AI and machine learning. So we are using different type of data throughout the pregnancy, birth, and newborn life to predict different neonatal outcomes. I will give you some example. So in this study, we applied the deep learning techniques to develop a metabolic health index. It has been showing very good performance in stratifying preterm children. In another study, we utilized a large-scale tabular data to perform longitudinal risk assessment. By linking maternal and neonatal data, we are able to monitor the progression of multiple disease throughout the children's birth. So from this example, we can see that machine learning has been used for predicting neonatal mobility. However, currently used data are just structural portion of the health data, such as tabular data and biomarkers. So the unstructured part of data, especially for clinical notes, is largely underutilized. So recently, as you know, that large language models have gained significant attention in healthcare. Through pre-training with large-scale corpus, they demonstrate a very good performance in natural language understanding. BERT model is one of the examples. It was originally developed by Google through pre-training with large-scale Wikipedia texts. Later, researchers further pre-trained BERT with domain-specific texts to develop BioBERT, clinical BERT, cancer BERT, and other domain-specific BERT model. So we are proposing neonatal BERT, a domain-specific BERT in neonatal mobility prediction. Similar to BERT model, training of a neonatal BERT also includes two stages, the pre-training stage and fine-tuning stage. After the development, the neonatal BERT was evaluated using two data sources. So here is the flowchart to show how we got the data. So the first data source is Stanford EHR database, including 32,000 newborns with more than 500,000 clinical notes for pre-training. And the second data source is the MIMIC and ICU database, including over 7,000 newborns. Then we use the clinical notes within the first day of birth to predict the future risk of neonatal mobilities. And we can see the model performance here. We have 18 different neonatal mobility outcomes here. The neonatal BERT show a very promising performance in terms of AU-ROC, AU-PRC, and F1 score compared to other baseline. So the baseline includes the model using tabular data and previous large language model, including BioBERT and bioclinical BERT. So the bold number indicate the best performance in the comparison. And this bar chart also give you a clear visualization that neonatal BERT outperform others. The red bar represent the neonatal BERT. So in the ASNO validation, neonatal BERT also achieved improved performance in predicting neonatal mortality in the MIMIC and ICU database compared to others, and prove its generalizability in different data sources. And then I will show you how neonatal BERT could potentially help in clinical practice. When baby is born, we will collect all data they have, including history and physical notes, delivery notes, and then we put them into the neonatal BERT model. Then we can predict the probability of different neonatal mobilities, and we can set the threshold to represent the risk level. Is a high risk or low risk or medium risk. Then we will, I think this panel will give the neonatologists a very clear understanding of the risk level for newborns, help them to determine the individual treatment, and also give some special attention to newborn with high risk. So in summary, in this study, we developed a large language model focusing on neonatal context. It achieves improved performance in 2 data sources. It also has a very strong potential to be used in many different tasks in neonatal care in the future. And so thank you very much for listening attention, and thanks to the lab, thanks MCHRI postdoc support reward for supporting this work, and welcome to reach out, and I'm happy to answer any question.
Really fascinating. I think these are exactly the kinds of tools that we are going to need. Thinking about this morning and trying to understand how maternal climate and exposure of a fetus may translate into those infant outcomes. And only through big data and the ability to understand big data are we going to be able to answer those questions.
Yes, very exciting work. Have you tested this on another group of patients? I'm always concerned about building a model on a selected group like Stanford patients, and then people say, well, we could try it on our patients. Have you taken another data set and tested this on other neonates that weren't from Stanford?
I think I can show you. So just to clarify, you mean selected group patient is it's a neonatal patient, or it's other like pediatrics, or? I think we actually test our neonatal birth model in another data set, or ESNO data set. It's from Mimic and ICU. Actually, this is from a very large hospital in Harvard. And as you see that from the performance, our neonatal birth also has a very good performance. And although this outcome is only included neonatal mortality due to the data availability, it also shows that our neonatal birth has the generalizability to like very different institution or other very different data sets.
Hi, John Klein from Adolescent Medicine. That was a really nice presentation. Thank you. In natural language processing, some of the concerns that have been raised is that they magnify some of the biases that might be there on the basis of racial or ethnic group because of what gets documented or how it gets documented. And I think there are some interesting challenges there with this kind of modeling, because some of the social determinants of health are also going to increase the risk of some of the outcomes. So I'm curious how or if you're going to be able to tease out some of that potential bias in the data because of the way that we interpret some of the findings into the chart when it comes to ethnicity or race.
Oh, thank you very much for the question. I totally understand that there might, machine learning model might be biased due to some, like you mentioned, that the social determinant of health issue. I think for now, this is still an open research area. Many researchers have developed different methods to deal with this problem. I think for now, I would recommend that if you want to deal with problem, we can do it at the data processing step. Like for example, we just we can manually or maybe use some algorithm to tease out all of the like social determinant of health or race or this type of demographic information, and then we use only like the history or some measurement to do this the natural language processing. But I think in the future, I believe that there will be more and more fancy method we can use like to deal with the problem to increase the AI fairness. Yeah, thank you.
Great. All very good good questions. Our last speaker is Lulu Xing. Lulu received her undergraduate degree in biomedicine from the University of Melbourne in Melbourne, Australia. In 2016, she was awarded a PhD degree in neuroscience from the Florey Institute of Neuroscience and Mental Health, University of Melbourne, where she identified a major underappreciated role for subventricular zone-derived neural precursor cells in the process of myelin regeneration after demyelinating injury. She developed a mouse model of conditional ablation, which could be adopted in different neurological contexts to further advance our knowledge and understanding of neurobiology. She joined the Pediatrics lab in October of 2002 as a research assistant, seeking to understand the role of OPCs in the development of glioma as tumor-initiating cells, and try to understand how they are regulated by complex interactions with several cell types within the tumor microenvironment. She uses a combination of transgenic mouse models, advanced imaging techniques, and single-cell biology approaches, really rounding out all of the tools we have here at Stanford. Lulu.
Thank you very much for your introduction. Good afternoon, and I really appreciate this opportunity to talk about my work today. So as you know, I'm interested in brain cancer, particularly pediatric low-grade gliomas, so which are tumors mainly driven by BRAF gene alterations. As you can see here from the pie chart, BRAF with 300E point mutation is the second most common mutation, accounting for 17% of all cases. The BRAF gene encodes a protein kinase involved in cell signaling pathways, and this BRAF point mutation can lead to continuous activation of the MAPK pathway, and leading to dysregulated asymmetric cell division and increased cell proliferation. This BRAF mutation is also prevalent in pleomorphic gentle astrocytoma and ganglioglioma. BRAF with 300E mutation can also be associated with tumor progression, particularly when combined with other oncogenic mutations like CDKN2A deletion, increasing the rate of tumor progression and aggressive tumor behavior. So therefore, high-grade gliomas with the BRAF mutation are very challenging to treat. My research is mainly focused on the role of glioma-associated oligodendrocyte progenitor cells, which are understudied. So we want to know how these non-mutated OPCs contribute to glioma progression. To understand this, we have generated query-dependent transgenic mice with different genotypes to mimic low-grade gliomas with either moderate rate or high rate of progression. The schematic diagram here shows how we can induce BRAF with 300E point mutation and CDKN2A deletion upon query-mediated recombination after adenoviral query injection. So you can see here that following adenoviral injection into the M1 cortex, we can monitor tumor progression over time using different analyses. And we hypothesize that glioma-associated OPCs are a significant driver of tumor plasticity and progression. But for today, I'm going to show you some preliminary data from the high-risk group only because these mice had poor survival outcomes compared to the mice from the moderate risk group. And I divide experimental time course into 3 time points at early, intermediate, and later stages of progression. Firstly, from the histological analysis, we found that our mouse model recapitulates the PXA like features with gliomas that can progress from low-grade to high-grade. And together with the immunohistochemical analysis, we found that at the early stage, there was a small focus of a tumor surrounded by OPCs, which are immunoreactive for PDGF receptor alpha. So you can see that OPCs start to proliferate and closely interact with the tumor cells. And then at the intermediate time point, these cells start to infiltrate into the tumor core, and there are more cells around the tumor border, suggesting that there was a formation of a tumorigenic niche. And also at the later time point, you can see that OPCs, there was a deeper OPC infiltration into the tumor core. And additionally, at higher magnification, you can see that these cells have a different morphology compared to distant OPCs outside the tumor core with bipolar morphology. So basically, there was a change in OPC morphology from bipolar, hyperramified, and then activated morphology. And additionally, from this graph, you can see that all pieces account for a significant proportion of glioma-associated infiltrating cells at intermediate and later stages of progression. So this suggests that all pieces might undergo dynamic functional changes during glioma progression. So in order to test this hypothesis, we are currently doing transcriptome profiling to determine whether these all pieces have distinct transcriptome profiles, which can be dependent on glioma stages and grades. We think that these all pieces may have an immunomodulatory function, so they may be involved in immune activation at the early stage. And then maybe later on they can be hijacked by tumor cells, so they become more immunosuppressive and secrete poor tumorigenic factors, cytokines to facilitate tumor progression. So overall, our low-grade glioma model sheds light on the critical roles of glioma-associated OPCs during progression, and therefore understanding these underlying mechanisms is vital for better treatment that can effectively manage pediatric glioma, because high-grade gliomas are very difficult to treat. So our research has important implications for pediatric glioma treatment, ultimately improving child health and outcomes for young patients with those challenging tumors. And lastly, I would like to thank people from the Portis Lab, especially my mentor, Associate Professor Claudia Portis, and my undergraduate student, Brandon Bui, for their support and contribution, and also people from the facilities for their help. And thank you very much for your attention. I'm happy to take any questions from the audience. Thank you.
Thank you, Lulu. That's very interesting. I had a question about, are there any theories of why the OPCs change morphology? I mean, we think about, pH or metabolism or things that, change proteins, but is there any theory of why that happens?
This is a very interesting question. I think that might indicate they may have interesting functions related to the morphology. For example, they may do at the beginning, they show the bipolar morphology, so that means they can be more migratory. But later on, they become more activated, so bigger like microglia, so they might be involved in phagocytosis or secretion of pro-inflammatory cytokines or chemokines to be involved in immunomodulation. So yeah, that's why we are doing single-cell work to validate this.
Great. So I thank you very much. If there are additional questions for our young, groundbreaking scientist, I hope that you can find them during the break. We'll ask that they all come to the front of the room to be easily discoverable. And I believe it is time for coffee. Is that right? No, it is not. That's in my mind. That's in my heart and my dream. Oh, I remember what it's time for. Exactly. Dr. Leonard, can I bring you up? Okay.
Yes.